All right, so today we are talking about DNA. And this week we'll be talking, we'll be beginning to talk about molecular biology. So some of this is review, depending on how much biology you've had in the past, and you know, it depends on how recently you had that biology as well, right? If you had biology last semester versus biology 10 years ago, it's all going to be relative to how recently you've studied it and how deep you went into it in Bio 1. So today we're talking about DNA and RNA. Pretty good detail. So DNA and RNA are both polymers, right? So polymers are long molecules made out of repeating units. And the repeating units in DNA and RNA both are called nucleotides. And so DNA and RNA have several things in common, but generally speaking, DNA and RNA are very different. They have different roles in the cell. They have different overall structures. But one of the things that they do have in common is that they're both polymers of nucleotides. All right, so let's talk about what a nucleotide is. It's composed of 3 parts. A nucleotide contains a base, A, T, G, C, or U, right? So this varies. It could be A, T, G, C, or U. We'll talk about what those are, what they look like in just a second. But every nucleotide contains a nitrogenous base, hence the name because of the ends, right? Every nucleotide also contains a pentose sugar. And again, it could be a hydroxypentose or a dehydroxypentose. I'll show you what that means in just a minute, and how to tell the difference between the hydroxypentose and the dehydroxypentose. And then every nucleotide also contains a phosphate group. There's nothing special about the phosphate group there. But you have to have all 3. You have to have the base, the sugar, and the phosphate group to have a nucleotide. Okay? Nucleotide with a T. Now, nucleosides, you're reading in your textbook, you might hear about these. Nucleosides don't have the phosphate group yet. Okay? They don't have that phosphate group. So which one is used as the monomer for DNA and RNA? Is it the nucleoside or the nucleotide? It's the nucleotide. Okay? So especially in your reading, if you see about nucleosides, that is not the polymer of DNA and RNA. Okay? It's the nucleotides. So they don't have that phosphate group yet. So DNA and RNA are made out of nucleotides. Okay? Nucleotides. So they have the base, which again varies depending on which one we're dealing with. They have the sugar, which again varies depending on if we're dealing with DNA or RNA. And then they have the phosphate, and the phosphate's the same in both DNA and RNA. All right, so let's talk about how the bases differ in DNA and RNA. DNA and RNA both have cytosine. That's a base that you'll find in DNA and RNA. DNA and RNA both have adenine, which we just represent with A, and cytosine we represent with C. Okay? DNA and RNA both have A, DNA and RNA both have C. DNA and RNA both have guanine, which we represent G. But one of the big distinguishing factors between DNA and RNA is that in DNA, thymine, T, is present, whereas in RNA, uracil is present. We represent with U. And these structurally are extremely similar. What's the only difference between them? You've just got this methyl group. That's the only difference. Okay? So structurally, they're extremely similar. The only difference is the thymine's got that methyl group, whereas the uracil does not. Okay, so DNA contains thymine, we represent with the capital T. RNA contains uracil, we represent with the uppercase U. You probably talked about some of this in Bio 1. I'm assuming you did. Yes? Okay. So, A, G, and C are in both DNA and RNA. In DNA, thymine, and in RNA, the thymine is replaced with uracil. Okay, let's talk about the sugar. Remember I told you there are differences in the sugar in DNA and RNA. Now we use these little primes, it looks like a little apostrophe, to distinguish the C's in the sugar from the C's in the bases. Okay, so if we're talking about the bases, we would just number them 1, 2, 3, 4, 5, 6, 7, 8, right? There's no primes. But because we've got both a sugar and a base, okay, that's why we use the prime. The prime goes for the carbons in the sugar, not the carbons in the base. So there would not be any primes if we were numbering these. There would be primes on the sugar. Okay, so you can separate them. So RNA contains ribose, and we begin numbering from the right, just like we have in all of our other sugars. Okay, so this is 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. There's ribose. Okay, now DNA contains deoxyribose. So if you've ever heard DNA, deoxyribonucleic acid, right? It contains deoxyribose. What's special about that? On carbon 2 prime, notice there's no OH group. That's a big deal, right? That's a big, big structural difference. So make sure you know the difference between ribose and deoxyribose, right? In the second carbon of the sugar, there's a hydroxyl, like you would expect on any sugar. Think back to last week, right? Every sugar is going to have an H and an OH on every carbon, unless you're talking about, ones like this, where they've got CH2OH. Whereas in DNA, it's just 2 hydrogens. There's no OH. That's a big, big, big difference. So ribose versus deoxyribose. Right? RNA stands for ribonucleic acid because it contains ribose, whereas DNA stands for deoxyribonucleic acid because it contains deoxyribose. Okay, so again, make sure you know how to number the carbons. We're numbering these carbons the same way we did last week when we were numbering the carbons in monosaccharides and polysaccharides, right? Starting here, 1, 2, 3, 4, 5. Be able to explain how the pentose sugar, the ribose in DNA differs from RNA, right? That's a big, 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 big difference. So if I gave you this is the one from a minute ago. Is this a nucleotide or a nucleoside? Side or tide, which one is it? It's a tide, right? It's got all 3. Is it from DNA or is it from RNA? It's from DNA. How do you know?
Because there's no OH on
Carbon 2 prime, right? Carbon 2 prime's right there, and it's deoxy. Good. Okay, this is something you probably talked about in Bio 1, Chargaff's observation. And it doesn't matter, this figure's from your textbook, what organism you analyze. The amount of A is equal to the amount of T, and the amount of G is equal to the amount of C. And it doesn't really matter which organism you pick. Those 2 are the same, and those 2 are the same. And so it led to the conclusion that DNA must be double-stranded. Right? The amount of one side there must be something complementary to the DNA. And we're going to come back to the double-stranded nature in a few minutes, but I just wanted to throw that out there now. This figure's from your textbook. So here's a summary. If we're going to compare DNA and RNA, what are some of the big differences that you need to know? You have to know this. Right? DNA is double-stranded. There are 2 strands. We're going to get into details of that in a few minutes. Whereas RNA is single-stranded. It's just 1 single strand. Okay? DNA is composed of A, T, G, and C, whereas RNA is composed of A, U, G, and C. Okay? In DNA, the sugar is deoxyribose, and in RNA, the sugar is ribose. So these are some of the major, major differences between DNA and RNA that you need to be able to recite to me, right? You need to know that DNA is double-stranded. You know that RNA is single-stranded. You need to know that everything's the same except thymine versus uracil in terms of the bases. And then in terms of the sugar, it's deoxyribose, meaning that that second carbon in the sugar has no OH group versus regular old street ribose, right? Nothing modified to ribose in RNA. What do they have in common? They're both polymers, right? Another thing they have in common is they're both made out of nucleotides instead of nucleosides. But otherwise, there's some pretty significant differences. What's the only difference between these 2? Right? There's only 1 methyl group difference. Structurally, thymine and uracil are very, very similar. So it's not like one's just, grossly different from the other. There's a methyl group separating these two. Not a humongous difference. Okay, so again, because these are polymers, right, they're repeating units. The repeating unit is the nucleotide. The nucleotides are linked together by what we call phosphodiester bonds. So how does that work? We're looking at the 3' OH group of your sugar, okay, and it's forming an ester to the phosphate group on the 5' carbon of the next nucleotide in line. I'll give you a second to jot that down. The 3' sugar OH and the 5' of the carbon next door's phosphate group. We call that a phosphodiester bond. I'll show you a picture in a minute. 3' OH group and the 5' phosphate group. It's a lot easier to see than it is to write it out. And all these figures, by the way, are in the textbook as well. 3' OH group and the 5' phosphate group. All right, so what you get here, remember we start numbering 1, 2, 3. Carbon number 1 is where your base is located. Carbon number 2 is where you tell if it's DNA or RNA, right? Here's carbon number 3 of the sugar, and there's its OH group, right? And then here's carbon number 5, right? 1, 2, 3, 4, 5. It's the phosphate of that carbon number 5. So these two pieces right here make our phosphodiester bond, okay? And so notice you've got one free phosphate that's ready to bond on this side to another monomer. You've got one free OH on this, right? So it doesn't matter if you've got 2 of them or 2 million of them. There's always going to be one end that's a free 5' phosphate, and there's always going to be one end that's a free 3' OH group, right? So no matter how many you put in the sequence, whether it's just 2 or whether it's a zillion, you're always going to have one end that's free on the top and one end that's free on the bottom, right? There's always going to be a free opening on both ends.
So at this point it mattered that the ones last week were 1, 4 bonds.
Yeah. So when you sequence DNA, what are you sequencing? The sugar and the phosphate, that's the same in all of the A, T, G, and C, right? You're going to have a free 5' end. You're going to have a free 3' end, no matter how many are in the chain. But as a convention, we always sequence from 5' to 3'. Because if I'm starting down here and you're starting up here, we're going to get different sequences, right? And when we sequence, we're not actually sequencing the sugar, because who cares? The sugar's the same in all of them. If it's DNA, they're all going to have the same sugar, right? What we're sequencing is the base that's sticking out. So if we had this little segment of DNA, we would sequence it from the 5' to the 3'. Always start at 5'. Okay, so that everybody's starting at the same place. Because if I start down here and you start down here, we're going to get different order. All right, so the sequence here would be A, C, G, T. Right, we're not sequencing this because this is the same in all the nucleotides. Right, what's different is the base. And this is probably something you did in Bio 1. Right? When you're sequencing DNA or you're doing practices with that, all you're doing here is looking at the base. Okay. And so this we call the sugar phosphate backbone, which again is not considered part of DNA sequencing. When you sequence DNA, you're just looking at the base. Okay? All you're doing is looking at the base. And then the phosphodiester bond, again, is occurring between the 3' OH group and the phosphate group of carbon 5. That's why there's always one 5' end that's open, and there's always one 3' end that's open, right? And again, if I gave you this, I could ask you, is it from DNA or RNA? Well, now there'd be 2 ways to tell, right? Because I've labeled it. If there's thymine, it obviously has to be from DNA. But maybe I don't label it. Maybe I just give you the bases and I didn't label it. You could still tell it's from DNA. How?
Well, not the base, because of the phosphate group.
Not because of the phosphate group. Look at the sugar. Right? How could you tell? Because RNA is going to have phosphate groups too. How could you tell it's from DNA? Look at carbon number 2. Right? So even if I didn't label what the name of the base was, if I just gave this to you and said, all right, is this a sequence from DNA or a sequence from RNA? Look at your sugar, and your sugar will tell you if it's DNA or RNA. Because DNA and RNA are both going to have phosphodiester linkages. What's going to be different is the sugar, right? And if I did label it, then obviously you could tell, yes, that's DNA. But again, remember, the difference between thymine and uracil is that methyl group's not there. That's the only difference. Okay, so Chargaff's observation concluded DNA must be double-stranded. Right? That means there are 2 strands, and something on one side must be complementary to something on the other side. Now, much later, when the structure of DNA was resolved, okay, much later, an interesting observation was revealed, okay? The 2 strands of DNA are not held together by covalent bonds. And this is really misleading because think about how all your textbooks draw DNA. They draw it as these ladder rungs, right? Which would imply that there are covalent bonds, right? That the 2 sides are bonded together. But they're actually not. The A and the T are linked together by 2 hydrogen bonds. The G and the C are linked together by 3 hydrogen bonds. So these are intermolecular forces holding the 2 strands of DNA together, not covalent bonds, which is very contrary to the way, you know, especially your average run-of-the-mill textbook represents a DNA molecule, right? It draws it as this, like it's a ladder. And that implies, oh man, there's this strong bond that's holding the 2 chains together. Well, yes, collectively, those hydrogen bonds add up to a lot. But a hydrogen bond doesn't light a candle in terms of bond strength to a covalent bond because it's not a bond, right? Remember, it's like a strong magnetic attraction, whereas a covalent bond is electrons being shared between nuclei, right? So hydrogen bonds are what's holding the 2 strands of DNA together, not covalent bonds, okay? It takes a lot more effort to break covalent bonds than it does to overcome hydrogen bonds. And this is one of the many reasons why DNA is so fragile. It's held together by hydrogen bonds. It's not held together by covalent bonds. The way you traditionally think about the way textbooks and even, you know, figures you see out in pop culture showing DNA as these like ladder rungs. And it's not. You shouldn't think of it like that. There's no covalent bond going on. Okay, so where's the hydrogen bonding? Here's your NH2, right? That's an amine that's capable of hydrogen bonding. It's hydrogen bonding with that ketone. And then that's an amine and that's an amine. So hydrogen bond here, hydrogen bond here between A and T. And then what do we have going on here? There's an amine capable of hydrogen bonding, right? There's an amine, hydrogen bonding, and then there's hydrogen bond between that amine and that ketone as well. Okay, so again, IMFs collectively are going to be strong, but they're not going to be anywhere near as, say, this bond strength, right? That's a covalent bond. This is an IMF. So if we were going to compare strength of this attraction versus strength of this covalent bond, this one kicks this one's butt all day long, right? Because this is not a transfer, this is not electrons being shared, whereas this is. And so, again, when you think about the way DNA is represented, it's usually represented as these solid lines, but don't think of it that way, right? Remember, IMFs are represented as dots, and that's really what it needs to be represented as. You need to think about it as dots between the 2 lungs, not these solid lines, okay? Make sense? Makes sense? And this is the classic picture you're probably familiar with, right? The textbook companies don't show all the sugars and the phosphates. They just represent this is nice, lovely ribbon. But in reality, it's a messy, clunky, blobs. It's not this nice, beautiful ribbon. So there's another textbook's representation. Hydrogen bond, hydrogen bond, hydrogen bond, right? But again, those should be dashes, not covalent bonds. That's a more accurate representation, right? So the two chains are called complementary, which means the sequence of one determines the sequence of the other. And you're responsible for knowing who is complementary to who. Okay, you need to know that A is complementary to T, which you probably still know from Bio 1, and G is complementary to C. And then the two chains run anti-parallel to each other. What does that mean? That means that one strand is going 5' to 3', right? And that's the one we sequence, because we sequence from 5' to 3'. So what would the complementary strand be? It's got to run 3' to 5', right? It's like on the highway, one's going north, one's going south, right? If this one's 5' to 3' and these two are complementary, then by definition, this one's got to be the opposite of this one. So this one's running north-south, for instance. This one's running south to north, right? If you've got the 5' in, which is, again, this is the one we sequence 5' to 3', you could fill in the other side, right?
So it's just the opposite.
And then these SP, right? That's the sugar phosphate backbone. So these are all covalent bonds. These are all covalent bonds. These are hydrogen bonds. And it's either 3 hydrogen bonds if we're talking about these guys, or 2 hydrogen bonds if we're talking about these guys. So why don't you give me the complementary sequence? If this is the strand on one side, you give me the other strand, and then label which is the 5' end and which is the 3' end. All right, let's go over what we had as our practice problem here. Remember, we sequence DNA 5' to 3', and if I am sequencing DNA, I'm not listing the sequence of both strands. I'm only listing the sequence of one strand, because if I know one strand, then I can fill in the other side if I needed it, right? So when I sequence DNA, I'll list it as single-stranded, even though I know in my mind it's actually double-stranded, right? If you know one side, then by default, you can figure out the other side. So if this strand is running 5' to 3', what's this strand running? It's running 3 to 5, right? And so if you've got A on one side, again, this is DNA, RNA single-stranded, so you wouldn't have a complementary strand in RNA. If you've got A on this side, what are you going to have here? T, and then C, and then A, G, G, T, T, A, G, right? And then again, you label this is the 3' end, this is the 5' end. Right? If you know one side, then by definition, you can figure out the other side. Okay, now this is just fun facts. I'm not going to make you memorize this for a test. To take a full turn, 360 degrees, that takes about 10 nucleotides and has a length of about 3.4 nanometers. So we're not talking about, a football field here, right? This is a small amount of space. And just a reminder that RNA is single-stranded, which means there's no complementary strand. And obviously, you can't have hydrogen bonds linking strands together that don't exist. If you only got one strand, you wouldn't have that hydrogen bonding to talk about, right? So let's talk about replication of DNA. Again, the amount of background you have in this is going to vary tremendously based on how much you talked about it when you took Bio 1 and how long ago you took Bio 1. Taking it right now versus 10 years ago, right? That's going to make a big difference. So before we get into DNA replication, let's remind ourselves what's the difference in a prokaryotic cell, what's the difference in a eukaryotic cell, right? Prokaryotes have no nucleus, so we're talking about bacteria, right? Whereas eukaryotes do have a nucleus, right? So we are composed of eukaryotic cells. So let's talk about prokaryotes and eukaryotes separately because their DNA is different. So bacteria, those guys are all prokaryotic, right? And their entire chromosome is a circular shape. I'm not going to make you memorize how many nucleotides it is, But you start at a beginning at the sequence called the replication origin, and then it moves bidirectionally. You get two at the end. Right, so here's a picture. You have a place where replication begins, and then it occurs both ways, and then you get two at the end. Now, bacterial DNA is really useful to us. We're going to talk about things we can do with bacterial DNA on Wednesday, but I'm just kind of throwing this out there now. Obviously, we're not bacteria, so our DNA replication is going to be different than, say, in a bacteria. So let's talk about what prokaryotes go through. Excuse me, prokaryotic chromosomes are just that single piece, right? And then it can be floating around in the cytoplasm because there's no nucleus. Now in eukaryotic chromosomes, right, the number of them varies, the sizes vary, and then the DNA is also supercoiled, but it's going to look different. So if this is our DNA, if you could take your DNA and stretch it out, let's say you take your entire DNA sequence and I stretched it out. Want to guess how long that molecule would be? If you could just pull it out of your cell and stretch it out to look at it. How long do you think it would be? Any guesses? There's a meter stick for reference. That's about not quite the length of that screen. Any guesses? It's kind of nasty meter stick because it sits on the chalkboard all day. Your DNA would measure 3 meters approximately, right? That's taller than the height of this room. There's 1 meter. There's 2 meters. So the height of this room is about 2 2/3 meters, right? So that's taller than this room. If you could take your DNA and stretch it out, it's approximately 3 meters. That's really long. How are you going to fit something 3 meters long inside each cell nucleus? And not just one of them, right? We have more than one chromosome. We'll talk more about that later, right? You got to do some major time condensing. So you take your double-stranded DNA, and then you wrap it around these little proteins called histones. Okay, so it wraps around these little histone proteins. This figure's in your book. Okay, so you have the histone, and then it wraps around that. And then those little histone beads condense around each other. So imagine if you took like a rope and you just start twisting it, right? Eventually it's going to coil on top of itself. You get this tight little, and then that condenses down even further, which we call chromatin. And so you can't really see this in the screen, but you can look at it in your book. It makes these little coils. Chromatin fiber condenses further down, which makes your chromosome. Okay, so you've got a 3 meter long molecule that's got to fit inside a cell nucleus. And you got to fit more than one chromosome in your cell nucleus. It's not like we're composed of one chromosome. Right? We'll talk about chromosomes and all that good stuff in a little bit. But I want you to get an appreciation for how much work it takes to fit this ginormous molecule inside not only a cell, but inside the cell nucleus. It's not like your whole cell is composed of just a chromosome and that's it, right? You've got to have a whole bunch of these inside your cells. And so this is called the karyotype. That's a picture of all your chromosomes. So we have 23 pairs, right? So each one of these is supercoiled DNA. Right? So what you're looking at here is not the DNA sequence right there. What you're looking at is chromatin that's been super condensed, right? When you're looking at those chromosomes. You're not actually looking at the DNA itself. I mean, you can think of it as the DNA, but there's a whole bunch of other stuff in there too. All right, so to replicate the DNA, you got to uncoil it, right? Because it's a big mess. Right? You've got to get down to the DNA itself. And I mean, the DNA, think about how awful it is to untangle a necklace that gets coiled up. Oh my gosh, I'd rather like throw it away sometimes, right? Unless it's like from your great-great-grandma and you're like, oh, okay, where's the microscope so I can, you know, just think how much it stinks to uncoil a tangled necklace, right? Okay, DNA is even worse, and it's going to get tangled. It's going to get tangled a lot, right? So if you don't untangle it, you can't replicate it, right? And then you're really sunk because the cell can't replicate if you don't make a copy of DNA. So we have to untangle it. And so there are proteins. Proteins do the job. Okay? Topoisomerase 1, topoisomerase 2 make cuts. They actually cut the DNA. And you think about that going, that's a little terrifying to think about, but your DNA gets chopped, right? And then those coils, those loops, whatever the knot looks like, can get passed through. And then other proteins actually piece it back together. Now you're going, oh, that's kind of scary. My DNA is being chopped up. Also think about the fact that proteins are just large molecules, right? They're not living things. I think a lot of students tend to think about DNA, replication, and cell processes as a whole as like, you know, little tiny machinery doing this stuff. Well, it is, but it's not like those are human machinery, like they have brains. These are all non-living things. A protein, you just put it in a beaker, right? It's a non-living thing. DNA itself is a non-living thing. So don't personify it, okay? Make sure you think about this as it's not living, right? A protein, not a living thing. DNA, not a living thing. It's a component of a living thing, but on its own, it's not like it's living. So here's the big picture. You got to unwind and untangle, right? Because it's going to get tangled up. And then the big picture is you're going to make 2 new strands. Right, if this is our original parent in purple, it's going to unwind, and then new sequences are going to come in. Now, does this just happen spontaneously? Do they just load in there? No. What kind of cellular machinery does this? It's unwound and untangled by proteins, and then these new ones are put in by proteins, and then it's put back together by proteins. Okay, so the cellular machinery that's doing all this is proteins. So you're going to get 2 strands out of it. And we call it semi-conservative. Semi-conservative. What does that mean? That means that the 2 strands that you get at the end of this have 1 strand that came directly from the parent and 1 strand that was newly synthesized. Okay, so in other words, you only are actually making 1 new strand. Why would that be advantageous? To only make 1 strand? Well, it's more effective, it's more efficient, yes. There's also a lower probability of making a mistake, right? Because if I have to take this and make a copy of the first strand and a copy of the second strand, I have doubled my probability of screwing up, right? Versus if I'm just making 1 new strand, I've only got the probability of screwing up once. Also, if I mess up on this one, would it affect this one? Right? These 2 are independent of each other. So if I make a boo-boo over here, that's not going to affect this one over here. So it's called semi-conservative because you take 1 parent strand and you don't do anything to it, and then the other one is the one that gets completely manufactured fresh. Okay, so that's why we call it semi-conservative. You're conserving 1 strand entirely intact, not doing anything to it. Okay, so that's what we mean by semi-conservative. And the place where this splitting begins, we call that replication fork. Now again, your textbook pictures are grossly oversimplifying this because this doesn't unwind without outside help, right? There's a big fat honking protein sitting right here that's doing the unwinding, okay? This doesn't just unwind spontaneously. And these don't just float in there, right? There are complex cellular machinery we call proteins that are putting them in place, okay? So, semi-conservative, you get 1 strand that's directly taken from the parent and 1 strand that's newly made. Okay, so there's a lot going into it, and I don't want you to think of it as this just simple little process that, oh, you know, it just splits and new stuff comes in and then we're done, right? It's not trivial. There's a lot that goes into it. So let's talk about some of those proteins that are involved. So what protein unwinds DNA? We call that helicase. Remember I mentioned when we were talking about cellulose last week, I said you don't have cellulase, right? ASE suffix is something we see a lot with proteins. So helicase, and an enzyme is the same thing as protein. Okay? Protein called helicase unwinds the DNA. Okay, you've got to do it correctly. And then we also talked about what protein is involved if you have a link, if you have a loop or a coil. I'm not going to get into the details of what topoisomerase 1 does versus what topoisomerase 2 does. Okay, save that for your full semester biochem. But those proteins do what? Topoisomerase 1 and 2, they remove those knots. Right? If you get a knot in your DNA, topoisomerase 1 versus topoisomerase 2. One makes a single-stranded cut, one makes a double-stranded cut. That's the bottom line. So helicase unwinds it. Topoisomerase 1/2 gets any of those knots out. And then where the DNA gets unwound is called the replication fork. So right here, where that happens. And again, imagine helicase parked right there, okay? Because that's what's actually doing this. So there's another figure. It's actually from a different textbook. Okay. And so there's a helicase, and then this protein, this is actually the parent strand, there's a topoisomerase sitting there. What do you think it's ready to do? It's getting rid of all of the knots, right? So helicase is the protein sitting here. This is a protein called primase. And what does it do? It puts a little piece on here called an RNA primer. Okay, so again, this all doesn't happen spontaneously. Cellular machinery is non-living. It doesn't know what to do, no, in quote marks, right? So you put this little primer here to help the party get started, okay? To tell the machinery, this is where we're starting. And these little primers are going to get chopped back out later. And then the polymerase, which we'll talk about in just a second, is what's actually making the new strand. So replication occurs 5' to 3', okay? One strand just flows continuously. But the other strand has to go backwards in chunks, 5' to 3'. Now I'm a lousy artist, but here is a nice picture of it. So you have your DNA that's, here it is, right? It's being unwound at the replication fork. Again, we're not showing any of the proteins here because it would just be too messy. So we've got one strand that's that's splitting apart, right? And replication occurs 5' to 3'. So this one just goes nice and smooth, right? Whereas this one, because of how it's put together, replication only occurs 5' to 3'. So you gotta have one that starts here and goes for a little bit. One that starts here that goes for a little bit, right? One that starts here that goes for a little bit. This one you can just have your machinery go along, nice and smooth, right? But as this unwinds, you have to do this way, and then you unwind a little bit more, and then you go this way, right? And then you unwind a little bit more, and then you go this way. So this is called the lagging strand because it's synthesized discontinuously, whereas this one is synthesized continuously. And I have a video that I'll show you in a few minutes that really helps put it into perspective. It kind of shows the big picture nicely. But then all these little pieces in the middle have to be synthesized. You have to smooth it all back out, right? It's a lot messier. All right, so this is what I just said, polymerase. And technically there's more than one type of polymerase. I'm not going to make you get into the details of that in an elementary biochem class, but you're going to just kind of look at the big picture here, right? Polymerases make new strand. And so here's another picture just showing that same idea. Now ligase is a protein that's going to smooth this back out, right? These little pieces, that's obviously not a continuous strand. So these all get filled in. A protein called ligase. Ligase is also a protein that smooths pieces back together. When topoisomerase chops the DNA, right? If topoisomerase comes through and cuts it, ligase has to put it back together, right? So ligase is a protein that seals strands back together after things have been cut. And then there are proofreader proteins, which we're not going to get into in great detail, but just know that, let's pretend an incorrect base is substituted here, which happens all the time, right? You have proofreader proteins. We're not going to worry about their names in elementary biochem, but just know mistakes happen all the time, right? And proofreading proteins do come along and check to make sure that the proper ones. Now, obviously, it's not going to catch every one of them, but there are proofreading proteins that are part of this picture too. So the whole process involves a lot of proteins, right? You've got your topoisomerase that's cutting it, getting rid of any links, any kinks, I should say, right? Helicase is unwinding. We're putting on a primer. Those primers have to be removed later by the protein primase, right? So that's kind of getting you started, getting you ready. Then polymerase is actually making the new strands. This one's going along continuously. This one's going along discontinuously, right? So ligase has to seal the pieces back together, right? So all of these proteins are playing a huge role here. And just remember, try really hard not to personify this too much, right? I always try to tell students, you know, you don't personify molecules in a beaker. You don't think about, oh, molecule A, molecule B, they're just reacting, right? You just think of them as these non-living things. Well, proteins are non-living things too. So don't think about this as something happening by a living cell. It's happening within a living cell, but the pieces themselves are not living. It's not like they have conscious decision-making skills, right? We can't think of them as, oh, let me think about what I need to do. But that's non-living. Okay, so let's talk about mutations, and that's where we'll end today. Mutations alter the sequence, okay? Now, they can occur from chemicals like mutagens. So if you ever heard something that's called mutagenic, that's what we're talking about. Radiation, and then again, some of those typos that proofreaders don't catch. And DNA makes proteins, and that's what DNA's job is. And so it'll produce a protein as a result that can be defective, or maybe it can be better. We don't know. Depends on what the protein's modification is and where it gets substituted. And so we'll talk more about this later, but again, a DNA mutation has an impact on the protein. Sometimes that protein is better as a result. Sometimes it's totally garbage as a result. So you can't just say all mutations are bad, right? That's the thing that I want you to think about. And there are 3 types of mutations that we're going to talk about in this class as an elementary biochem class. A point mutation is where you put just a single wrong base in. Okay, so for instance, if you say the sequence should be ATG, GAC, TTC. Okay, instead of having a G there, a C gets substituted there. And it now becomes ATG, CAC, TTC. That's an example of a point mutation, right? C gets substituted for a G. Or maybe an A gets substituted for a G, right? Something other than G gets put in right there. That's a point mutation. Point mutation. Okay, a deletion mutation is where you lose a nucleotide, at least one, maybe more. Right? So if this is what the DNA is supposed to be, ATG, GAC, TTC, okay, that GAC is gone. You've completely lost it. And now it's just ATG, TTC. Right? That's a big time deletion. You've lost 3 bases. That's probably bad news for your protein. And we'll see this next week when we look at protein sequencing in great detail. And an insertion mutation, as the name implies, is where one or more nucleotides get shoved in where it shouldn't go. Okay, so if this is the normal DNA sequence, ATG, GAC, TTC, now it becomes ATG, and this CTC got smushed in there, right there. All right, so now it's 3 bases longer, which, like I said about the deletion, it's going to be bad news for your protein. Because when we talk about protein synthesis is read in triplicate, in 3s, right? So it's going to shift your reading frame significantly with both deletions and insertions. So those tend to be pretty bad news. Okay, do you understand the difference between point, deletion, and insertion? And those are the only 3 mutations that we're going to worry about as elementary biochem students. So something else that can happen during radiation exposure is you can have what's called photodimerization. And so instead of your DNA having these nice complementary hydrogen bonds, these 2 actually covalently bond to each other. Oh boy, that's really bad news, right? Because now we don't have that nice complementary hydrogen bonding occurring. We've actually got a covalent bond between two T's, covalent bond between two A's. That's really bad, really, really bad news. And so this is a word you may have heard before, the telomere or the telomere. That's the end of the DNA strand, very end. And the ends are not fully replicated, the ends of the DNA. And so one of the things that's good about this is DNA can't just replicate forever and ever and ever and ever and ever, right? If the ends are not fully replicated. Because if every time you replicate DNA, the end couple of bases gets chopped off, eventually the DNA is not functional anymore, right? So that means that that DNA's got a lifespan. And that would be a good thing because you have these errors that happen and all that kind of stuff. And so some, I say some because not all, some cancerous cells have shown to have this protein called telomerase, which can actually restore the telomere. And that's one of the theories. Again, we have to be really careful when we're talking about cancerous cells because there's so many different types. But one of the theories on why cancerous cells can just replicate forever is because they have the ability to put those telomeres back on, right? Now, I have to say some and not all, because are all cancers the same? No, of course not, right? So that's one theory about why cancerous cells can just divide forever, because they have the ability to stick that telomere back on. Now, again, this is just one out of many theories on some cancers. Have to have a big disclaimer when we're talking about cancer cells. So that's where we're going to stop in terms of what I have. Do you have any questions or things we want to talk about before we wrap up?
Actually, yeah.
Okay.
Telomerase, is
Mhm.
that on RNA too, or just DNA?
It would just be on DNA. Because RNA gets made for short-term use, and then it's disintegrated pretty much right away. Yeah. I'm actually going to stop the recording here. I've got about a 5-minute video I want to show you, which is copyrighted, which is why I can't have it on the video. So stop the video.